Our next presenter, and the last presenter, not the how do they say, save the best for last. Our next speaker is Chinenye Nnajide, and she is going to be presenting on using DNA sequencing to trace the emergence of Colibacillosis in Saskatchewan broiler chicken farms. I love a good field study, so I'm very excited to hear this. So Chinenye, please go ahead.
Okay. Can everyone see my slides?
Yeah, you're great. Go ahead.
Okay. Good afternoon, everyone. Thanks, Brenda, for that wonderful introduction. It's a great pleasure to meet all of us virtually, and I believe or hope that I will meet some of us sometime in the nearest future in person. My name is Chinenye Nnajide, preferably Rosemary. I'm a PhD student in departments of biochemistry, microbiology, and immunology at the University of Saskatchewan, working under the supervision of Dr. Aaron White. And today I would want to share my research on using DNA sequencing to trace the emergence of Colibacillosis on Saskatchewan broiler chickens. But before I proceed, I would want to give a very brief background on what Colibacillosis is all about. Colibacillosis is a very broad term that refers to infectious diseases caused by pathogenic E. coli. But for the purpose of my research, I'll be focusing on Colibacillosis in chickens, and these are multisystemic or localized infections that are caused by avian pathogenic E. coli. This happens to be #1 cause of mortality and morbidity in poultry industry worldwide, and it has caused a whole lot of economic losses, which has gotten as high as over $100 billion in the poultry industry worldwide. And only in US, it has caused about $40 million losses as a result of loss of carcasses and cost of treatment of these diseases. Now, there are a lot of things that are yet to be known about these apex strains that causes Colibacillosis in chickens. #1 is that they are very diverse in the sense that it is very hard to find a same strain that has this set of virulent factors. 2 is that studies have also reported that these apex strains can colonize the gastrointestinal tracts of chickens without actually causing any infection or any disease. But then in the presence of stressors, they happen to translocate to extraintestinal sites to cause Colibacillosis in chickens. Also, a lot of studies have also isolated apex strains from chickens that happens to share same genetic makeup with E. coli strains that causes human infections like urinary tract infections, neonatal meningitis, and the rest of them. Now, this is what normally how this Colibacillosis is diagnosed. First of all, producers send their chickens to poultry extension teams at the University of Saskatchewan, and then they happen to pre-diagnose for Colibacillosis, and then they send it to prior diagnostic studies to confirm that these actually chickens that died as a result of Colibacillosis. Then they use this monitor, which is an automated system, to confirm that they are that they are Colibacillosis cases. Then after which the reports are sent back to producers without actually further characterizing these E. coli strains. They just throw away the E. coli strains without any further characterization. Now we decided to embark on this study to further characterize these E. coli strains to know what actually makes them to cause Colibacillosis, to characterize them phenotypically and also genotypically. Then what do we do? Oh, sorry. Oh, my slide can't move again. Okay, sorry. Now this is an overview of my project. Initially, we actually designed this project to just compare two populations. First, the diseased birds that died as a result of Colibacillosis, and then the healthy chickens from same flock. But then this first part of the project was conducted or carried out by a master student that happens to have graduated. Then her result shows that these two populations, E. coli from these two populations are not similar at all, in the sense that E. coli strains from healthy chickens are not same with E. coli strains that causes diseases. So my project now entered, we had to embark on my part of my project. We actually hypothesized that environmental sites could harbor these apex strains that causes Colibacillosis. First, we actually noted the sites that could harbor apex strains. We noted 8 sites, and then we collected samples from these 8 sites. First, you could see from this overview, first we isolate E. coli strains from these samples, and then we phenotypically characterize them by conducting the antimicrobial resistance profile checking their ability to produce coli and cellulose, which helps them to produce biofilm, and then check the motility and the rest of them. So we have decided that the barn environment can serve as a reservoir for apex strains circulating in Saskatchewan broiler farms. Also, we have decided that resistant and virulent genes can be transferred between different E. coli strains circulating in the broiler farm environment. So for my objective, first, we intend to isolate and characterize E. coli strains from this barn environment and then also generate their whole genome sequences. Second, we intend to characterize and also phenotypically check the presence of some ability of them to cause diseases in chickens. Then finally, we intend to compare these phenotypes and genotypes of these 3 E. coli populations. So what do we do? For the first objective, we isolate E. coli from farm environment. This is an example of feed sample that was collected by the poultry extension team. They send it down to our lab. Then we get this feed sample, homogenize them in PBS, and then played out on McConkey agar, which helps to isolate lactose fermenters and the rest of them. Then when we get these E. coli strains, we subculture to get a single colony and then confirm that E. coli strains by conducting the indole and citrate tests, which will be positive and negative. Then after we are done with this part of the project, we go ahead to select the strains that we want to sequence in order to avoid selecting same strains, because these are E. coli strains from the environment. Basically, we have a lot of E. coli strains circulating. We carry out we check the ability to produce coli and cellulose, and thus is what we basically do. We grow them on Congo red and Kako floor white agar, and then to differentiate different strains, and then after which we get them stored at 20% glycerol and then send them to -80 freezer. Then for the whole genome sequencing, we use 2 different techniques. First is the nanopore. This helps us to generate long reads. And then the second is the Illumina sequencing, which helps us to generate the short reads. So these short reads, basically when we want to draft the whole genomes, first we use the short reads. Then we generate this we generate the short reads, and then using the long reads, we stitch up the short reads in order to get the entire genome. So for the second objective, what we do is first being able to carry out the antimicrobial resistance testing. Here we use we streak out those isolates on blood agar. Then we send them to one of the collaborators' lab, which happens to be Dr. Joe Rubin's. And then in the lab, they use the NEGMI 42, which is an automated system to test for the ability to resist antimicrobials. Then for the second phenotypic testing that we've done, we use the crystal violence assay to check the ability of those strains to produce biofilm, because these are environmental strains. So basically, some of them should produce biofilm in order that helps them to resist harsh environment in the environment, harsh weather in the environment and all that. So for the third test we do, we actually do the motility test, which helps them to invade the intestines and deeper tissues to cause this colibacillosis. How we did this was we just grew up this isolates on LB broth overnight, and then we spot them. We standardize the culture at 1.0, and then we spot them on soft agar, and then incubate at 37 degrees Celsius for 18 hours. Then after the 18 hours, we view the rings to see or view their ability to move or migrate from the spot where they were inoculated to further path. So my results so far, we've not really actually done much except for the molecular part, which happens to be just generating the library and sequencing. So far, we've been able to collect a total of 119 samples from 8 different farm environments. And then we've been able to get 216 E. coli strains from these sites. And then for where we are presently, for the first part, like I said earlier, this first 2 parts was actually conducted by a master's student that graduated. So the results she got showed that these E. coli strains were 2 different populations. So far, she was able to isolate 503 E. coli strains. And we've already sequenced 105 strains, And then for the healthy E. coli strains, that's E. coli strain from the healthy birds, she isolated 149 E. coli strains, and we've been able to sequence 75 strains. Then for the part of my project, but invariably, I was able to also help in sequencing this part that she wasn't able to complete. Then for environmental E. coli strains, we've been able to isolate 216 E. coli strains, and our goal is to sequence 120 E. coli strains from this site. So the summary of my E. coli whole genome sequences for the environmental strains, we've been able to, like I said earlier, get 216 strains. And then for nanopore sequencing, which generates the long reads, we've been able to generate 108 whole genome sequences of those E. coli strains. And then for linear sequencing, we've been able to do 76 E. coli strains using sequenced E. coli strains using linear sequencing. Then for my antimicrobial resistant testing, I highlighted the part that I actually did, which was the environmental strains. You could see from here that these disease-causing E. coli strains are actually more resistant in comparison with the fecal that's from healthy and then environment. This shows that invariably, those two populations are different, and this actually different. So in total, we have 20 out of 66 E. coli strains that showed resistance to tetracycline. And approximately 65% of the environmental E. coli strains are susceptible to all antibiotics that were tested. Then for my biofilm formation quantitation using the CVRC, this is the part as well that I did. And then the first part was actually conducted by her. You could see from here that those strains actually grew more biofilm in nutrient-rich media, which is the BHI. And then for those, for the Kikau, it's actually the opposite. And for the systemic E. coli strain, that's from diseased birds, it's similar to this. There's no definite pattern at which those E. coli strains grow biofilm in this media. And this is what we intend to look into, to check the reason why some of them produce biofilm in nutrient-rich media, but then don't produce in nutrient minimal media. And also we intend to also check for gene expressions that could help them to actually grow biofilm in this media. So in summary, like I said earlier, disease-causing E. coli strains are more resistant, antimicrobial resistant than the healthy E. coli strains and then environmental E. coli strains. Also, a whole lot of diversity, and there's no absolute patterns at which this occur. For the biofilm formation, E. coli strains from one group form stronger biofilm in biofilm-rich media, which we intend to look into to check, to know the reason why this happens, because irrespective of the type of media, they should be able to produce biofilm in all the media. Then for the whole genome sequencing, we've been able to sequence 120 E. coli strains, 105 disease-causing E. coli strains, and 75 E. coli from healthy chickens. We also hope to complete this whole genome sequencing and comparing those three populations by September, which will be done with the help of our postdoc, Dr. Haley Sanderson. And in future, I intend to incorporate a whole lot of phenotypic characterization in vitro to check what helps these environmental strains to survive in the environment. And at the end, if we are able to find a particular gene that are present in disease-causing E. coli strains, we could try to carry out chicken infection experiments by knocking out those genes that cause Colibacillosis and then test them in healthy chickens. Then at the end, if the chickens happens not to be sick, we could use that as a goal to to find a prevention for this Colibacillosis. We could incorporate the use of anti-virulent factors, the use of antibiofilms, and the use of all naturally occurring organic oils that could help them to prevent the attachment and adhesion on those tissues. Then they won't be able to cause Colibacillosis in chickens. I would like to acknowledge my supervisor, Dr. Aaron White, my lab members, most especially Madeline McCarthy, for carrying out the first part of this experiment, Dr. Haley Sanderson for helping us out with the bioinformatics part of the project, Dr. Steve Varangi for helping out with some of the phenotypic testing and also guiding us in most of the protocols that we need to carry out. Then I would like to thank the Poultry Extension team for helping me out with the sample collection my collaborators, then fund the agencies, Saskatchewan Chicken Farmers, NSAC, Video Intervac, and Government of Saskatchewan. Thank you very much. If you have any questions, I'll be happy to take them.
Thank you very much, Chinenye. Unfortunately, we are past our time and won't be able to do any questions, but thank you very much for the very informative presentation and for doing this work. Like I said, I'm not a lab person at all, but I'm very appreciative for the people out there that enjoy it and love it and are helping to move our industry forward. So thank you very much for your presentation, and I wish you all the best with your research as you go forward. And with that, folks, we are concluding the second annual Canadian Poultry Research Forum. We're so grateful for all of our presenters, our attendees and sponsors for the great questions and interaction that we've had over the last 3 days. I can't believe it's over. It happened so quickly. On behalf of the whole planning committee and our greater research community, I would like to thank once again our sponsors whose funding is the reason that this event is able to carry forward. So our presenting sponsor is the Poultry Innovation Partnership, and our gold level sponsors are Chicken Farmers of Saskatchewan, Food from Thought in Guelph, and the Egg Farmers of Canada. On the silver level, thank you to Burnbrae, Canadian Hatching Egg Producers, Ritchie Smith Feeds, and Turkey Farmers of Canada. And our partners are the Poultry Industry Council and Canadian Poultry Magazine. So once again, thank you so much for being part of the forum, for celebrating and showcasing Canadian poultry research. We've had several questions about the recordings. We will be uploading those sessions into the virtual event platform over the next few days, and we hope to have that completed by early next week. We'd love to help you, to have you help us develop the program and format for future forums. So you can watch your email for a post-event survey that will be sent. I'm actually not sure when it'll be sent. Just watch your email, I guess is the best way of saying that. So with that, thank you so much for being a part of the event over the last 3 days. And I wish you all a very pleasant rest of your day. Thank you.